So, folks, in this presentation, we're going to be talking about probably one of the most incredible molecules discovered in the past 100 years. And that molecule is deoxyribonucleic acid, DNA. We're going to talk about the history of that discovery, the structure of DNA, and also how DNA replicates, which really means how it works. I started off with this picture here of the Human Genome Project. A genome is all the DNA that makes up a particular organism. So that is basically the instruction manual for that organism. In the case of humans, we have 46 chromosomes that make up our genome, 2 sets of 23, and all of these chromosomes contain all the genetic information you need to create a human. So just a quick review, DNA is found in the nucleus of eukaryotic cells. It can be found in the form of chromosomes during cell division. And if you unravel that chromosome, the DNA is just really a tightly wound, or a chromosome is just really tightly wound DNA around protein. And if you unravel that, keep unraveling, you end up with this molecule called DNA. So DNA structure, pretty big deal. In the 1900s, it really changed, and even now, it really changed the way we do things. And here are just a few examples to give you some context of the importance of this molecule. Here's an old-time magazine, and the curiosity here is, will there ever be another you? And what they're referring to is this sheep that was actually cloned. And that's really thanks to us discovering the structure of DNA and exactly how it works. We were able to actually clone a sheep and other animals since. Other things we're able to do through genetic engineering is certain animals have instructions in their DNA that code for proteins that allow them to glow. It's called bioluminescence. And we've actually taken some of those genes and put them in other animals like these mice, and as a result, they are able to glow. Another headliner is these things called GMOs, genetically modified organisms. And GMOs are really a controversial subject, but really what we're doing in the case of this tomato and many other plants that we grow for agriculture is making them bigger, tastier, well, maybe tastier, and redder, more sellable, stronger, and we do this with animals too, and there's a lot of question marks as to the effects, the long-term effects of doing this to organisms that we consume. Another huge part of how DNA is affected the way we do things now is this thing called gene therapy, where we can actually identify genes that aren't working properly in a human and replace them with genes that do work properly. So pretty big steps. We're actually able to save, in some cases, people from having to live with a terrible genetic disease. And just to make it interesting, if you recall Star Wars, all of these individuals are actually clones of the same person. So kind of interesting. So the history of DNA. There are many scientists that contributed to our understanding of DNA's function and structure, and this took a very long time, and really it is a great example of part of the history of human thinking and the scientific method, because it's really a long series of observation, questioning observation, and then creating hypotheses, testing those hypotheses, and referring to the work of scientists before. So I'm just going to mention a few of these scientists, and we'll kind of have a quick synopsis of what they did. So Gregor Mendel is our first and oldest. He's actually was an Austrian monk in the 1800s. Frederick Griffith and Oswald Avery, they're in the 1900s, around the 1920s and 40s. Erwin Chargaff, Rosalind Franklin, and Maurice Wilkins. And finally, the structure was really nailed down by Watson and Crick. So if we put them into a timeline here, we can take a quick look at how all this kind of lays out and just how long it took to really start to get this. And we've gone much beyond this, but this is really the basics of how we got to our understanding of DNA structure. So 1857 to 1953, so not that long ago, except for 1857, quite a while ago, but 1953, not so long ago. We start with this Austrian monk, Gregor Mendel. And Gregor Mendel studied heredity in pea plants mainly. And he wanted to know, well, how do the traits organisms have get passed through each generation? And why do some traits appear and reappear? And we often see this with things like eye color. Like, why do I have blue eyes and my parents don't? Or why does one of my parents have brown eyes and one have blue and I have brown? Or something like that. These are the kind of questions he was trying to answer. So he didn't know what DNA was. He knew there was something in organisms that allowed them to pass on traits and information. We didn't know what it was. And he studied basically only what he could see by breeding plants with his own eyes. Many years later, Frederick Griffith comes along in 1928, and Griffith is trying to actually study pneumonia because it was killing people at the time. It's caused by a bacteria. And what he figured out was, through a series of experiments, if he put dead heat-killed bacteria that had DNA codes, he didn't know about DNA, though he called it a factor, for being deadly with harmless bacteria, somehow the harmless bacteria would transform and become deadly. So there was some kind of informational exchange between the deadly bacteria that were dead and the living harmless bacteria which turned deadly. He didn't know that DNA was doing this, but he did call this a factor. Some factor is carrying this information from one organism to the other. Oswald Avery, on the other hand, in 1943, worked off of Frederick Griffith's work in a way, and he and his team determined that, yes, actually, DNA is the molecule that's carrying this information. So it was kind of like the factor that that Griffith was talking about. And he basically used enzymes to destroy any carbohydrates, proteins, or lipids, and found that when all of those things were destroyed, transformation still occurred, and therefore DNA must be the molecule that is transmitting the information from one bacteria to the next, or bacterium. So later on, Erwin Chargaff studies DNA further, and he finds out that DNA has these nucleotide bases that occur in specific patterns. And that pattern shows that, and these are the names of the bases, which we'll talk about later, show that adenine and thymine typically occurred in the same frequency, and guanine and cytosine. And he actually developed what's called Chargaff's rules, that adenine and thymine are always together, and guanine and cytosine are always together. And we'll talk much more about these, very important. We'll talk about how they work in the molecule of DNA. Chargaff didn't really know entirely, but he did establish this rule. Rosalind Franklin and Maurice Wilkins in 1951 used a technique called X-ray diffraction, where they would use X-rays through a crystallized sample of DNA, and they started to see this helical shape. So at the time, people were like, okay, well, we know there's these adenine and guanine. We know that DNA carries information. Well, what exactly does this molecule look like? And what they figured out was that DNA has this helical shape. They didn't know a whole ton of detail, but they knew it was like this helix shape. Finally, a little while later, Watson and Crick are really the ones to be credited for the discovery of the structure of DNA, and they were the first to build a model of the DNA molecule and show how exactly it looked, and that was the double helix. It kind of went into history that they were the discoverers. However, Rosalind Franklin's work, she passed away before it could really be published. It was actually published after 1953, and so she didn't really get all the credit she probably should have for her work, which Watson and Crick relied pretty heavily on to make their discovery. So she was really a footnote in their paper and probably should have been more, but she was unable to be awarded the Nobel Prize because unfortunately she had passed away before her work was really recognized. So Watson and Crick are known as the ones who really put it all together and finally made that first molecule, or that first model of DNA, which was really opened the door for tons and tons and tons of researchers. So the next piece here, we want to, okay, Watson Crick discovered the structure of DNA. Well, what exactly does it look like? So we want to describe the structure of a nucleotide, which is basically a unit of DNA or a piece of DNA. There are 4 of them in DNA. And we want to describe how nucleic acids are made of long chains of nucleotides. So really, what does DNA look like molecularly? So DNA and chromosomes are kind of synonymous. DNA makes up chromosomes. Chromosomes have some protein in them too. But really, chromosomes are just tightly wrapped DNA, if you remember back to that first diagram. So prokaryotes, or bacteria really, lack a nucleus. DNA is just in the cytoplasm, and that's what you can see here in this diagram. The DNA is just sitting in the cytoplasm, no nucleus in this bacterium. And their chromosome is a single circular molecule of DNA, and it kind of looks like this, just a circle. So when they divide, they make 2 copies of this, and they split them apart. So that's DNA in a prokaryote. DNA and chromosomes in a eukaryote, however, are a little bit different. Eukaryotes, or eukaryotic cells, are a little more complex, and they have DNA housed in a nucleus, which I think we've all seen. Each eukaryotic cell, depending on what organism it belongs to, has a specific number of chromosomes in it, which varies between species. In the case of humans, guess it. Okay, 46. Hopefully you knew that. 46 chromosomes in humans. Other organisms have fewer or more. We don't fully understand why, but that's our number right there. 2 sets of 23. And our chromosomes are essentially made of DNA. If you unravel them, you'll see this double helix molecule discovered by Watson and Crick and other scientists. So the discovery of DNA, mainly credited to Watson and Crick. They won the Nobel Prize and also were the first to publish a paper describing the structure, and there's them standing with that first model. And you can see, it's kind of hard to tell, but you can see the double helix that they're trying to show you. Theirs is a little more complex. So this structure explains how the molecule can be replicated. That's the key, because remember, we've known that this molecule carries genetic information, but how exactly does it work? And what does it do to carry this information? So here are our bases again, and we're going to get more into them. And you can see this double strand, and we'll talk more about what this whole diagram means and what's happening here as we go. So just a quick review before we get into the structure. So monomers and polymers. Many of our organic compounds have them. Proteins, carbohydrates. We have proteins with amino acids, and we have carbohydrates with we can have glucose monomers or different sugar monomers. In the case of DNA, we have these things called nucleotides. And just to review, a monomer is just a subunit molecule. So I'll say these are monomers here. We'll put an M on there. And basically, if I want to join these together to make a bigger molecule, I'll create some type of bond, and that bond will cause these smaller individual molecules to become one larger one, called a polymer. And so that's a quick review for you. Remember, we're talking about nucleic acids. They're the ones that are carrying genetic information. Others are very important, but this is what we're focusing on. So the structure of DNA. So DNA, like I said, is made of monomers called nucleotides. And we just want to get a basic idea for what these nucleotides have in them. So each nucleotide is made of 5-carbon sugar. There's our 5-carbon sugar. 5 carbons, we got a C there, probably here, there, carbon, carbon. We have a phosphate group, that's here. We'll make that a big P. So there's our phosphate group. And finally we have our nitrogenous base right here. And the base is one of those A's, G's, T's, or C's in DNA. So sugar phosphate backbone. So this is the outer structure of the DNA molecule. So by sugar phosphate backbone, I'm referring to this blue part here. And if we zoom in on it and kind of flatten the DNA out, it's this here. So there's our sugar, S, and there's our phosphate. Another sugar and a phosphate. So that's kind of what we call the backbone of DNA, and that's really important for the structure of DNA. And this needs to be very strong, because if you think about it, if this is the instructions for an organism, it needs to be very stable so that no changes get made by accident, because that could hurt the organism. So it's composed of a 5-carbon sugar, we saw that, and that sugar is deoxyribose. Remember, ose, that means sugar, and a phosphate group, and there they are again, SP. So continuing with that, the sugar molecules bond to the nitrogenous bases. So here's our sugar, here's our nitrogenous base. So NB, nitrogenous bases right there. And the phosphate groups hold the sugars together. So there's our phosphate group holding it to the sugar. Again, there's our phosphate holding to the sugar. And you can see how they hold the sugars together. On the other hand, you have the other part of the deoxyribose, or the sugar, hanging on to our nitrogenous base there. So you can see kind of how these get these monomers. Here's our monomer right here, our nucleotide. See how they all get snapped together to make a big polynucleotide, which is just a polymer. We call that DNA when we put them all together. So nitrogenous bases. So here's really kind of, these are kind of the words in the book of DNA. So molecules that bond the two complementary strands of DNA together. So by two complementary strands, I mean one side is a strand here, one is a strand here. So they're kind of complemented because they stick together in a certain way. So they're held together between strands by these weak hydrogen bonds. So in here there's a hydrogen bond, and if you recall, those are the same kind of bonds that form between water molecules, and there's a reason these are weak. Because DNA needs to be strong here, like we said, but these actually need to open up. So they're weak bonds so that we can kind of open DNA up, and it's kind of like opening a book. We want to be able to look inside this book and make copies of it so that we can instruct an organism on what proteins to make. And that's what we mean by instruction manual when we talk about analogies for DNA. So here's a complementary side here, and here's a complementary side here. And we'll talk more about this because this is really what allows DNA to make exact copies of itself for cell division and growth. So how exactly do these nitrogenous bases work? Well, here's how they work. The language of genetics is really a 4-letter alphabet. A, T, G, and C. And those refer to adenine, thymine, guanine, and cytosine. And these bond in a particular way, which means they can make like a code, and we call this the genetic code. And that code can be interpreted by enzymes and by your body to make different structures. So this is where DNA it's so amazing that we discovered this, because this is what is telling us what DNA is coding for and how it kind of works. So let's dig in here a little bit. If you look into this molecule, here's our backbone again, and inside are these nitrogenous bases, and the bases are T's and A's bond together, and A and T, again, same thing, and then G and C bond together, and also G and C bond here. So adenine will always bond with thymine, so A and T, and guanine will always bond with cytosine. And this is really what allows DNA to retain that code that everybody talks about, the genetic code. And that's happening in the bases here in the center. So it's important to remember this because in order to understand and interpret DNA, you have to understand kind of how it works. And really the basics of how it works is you always have the same bonding pattern between the strands depending on the base that's there. So whenever you have guanine, that bonds with cytosine. Whenever you have adenine, that bonds with thymine. And here's how you can kind of see they bond together to kind of form the rungs of the ladder. And here's our phosphate sugar deoxyribose backbone here, which doesn't really change. So, okay, we looked at the history of the discovery of DNA and all the scientists that helped contribute. We've looked at the basic structure. DNA is made of these things called nucleotides. They string together to make a polynucleotide, which is basically just a big DNA molecule. And there's these things called nitrogenous bases that bond in particular patterns to kind of make a code. And there's 2 strands to each molecule, and they twist to form a double helix. Well, how does all that work? How does all that make replication? In other words, how do we start with one person, or 2 in the case of humans, and then they have offspring that look pretty similar? How do we make copies of an organism, or how does DNA make copies to make copies of an organism? So that's kind of where we're going with this. So DNA replication. Each strand of the double helix has information to construct the other. We call this complementary. So by complementary, I mean if you pull DNA apart and you have one strand here and one strand here, based on the code in these nucleotides, you can actually construct another full, complete molecule of DNA by just adding the pieces back that are required, or the bases back. And that's what's so amazing. We have these complementary strands. You can open this book and easily copy it based on the template or the complementary strand. So this process occurs before cell divides and is called replication. So if you zoom in a lot into a nucleus, before the chromosomes show up in prophase, all the DNA is actually copied. And that's why when we look at a cell that's already in mitosis, it has these 2 identical strands. So same exact lettering on each one, so same genes on each side. And later on they'll split apart, so our final daughter cell will look like this, with just a chromosome that's not duplicated. So that's just one chromosome there that's not duplicated. This is also one chromosome that is duplicated, and these will split apart later during anaphase. So the important thing to know is, though, that all the DNA gets replicated in a cell before it divides, and that's why you see chromosomes with an extra copy attached with the centromere there. So during DNA replication, basically the DNA opens up kind of like a book. We're going to talk more about this. And those complementary strands are used to help make another copy, so that at the end of this whole process you end up with 2 exact molecules. And the reason that's possible is because of these bonds that are occurring between in a specific way. Remember, G's with C's, T's with A's, in between the bases of the DNA molecule. So once again, you can see, remembering back to those hydrogen bonds, which are represented by these little dots, you can see where the DNA has to open up to be used, and that's why those bonds need to be weak, so that it's easily opened up and then closed, and that's why these hydrogen bonds occur there. So kind of interesting. Same bond that's occurring in water is occurring in your DNA. So just another look at this. So the process that allows the daughter cell to have an exact copy of the parent's cell. So remember, if DNA is replicating, it's making an exact copy. So this chromosome we started with here will duplicate, and we'll go through the whole division process, and in that final cell, there it is again. So remember, they just separate. So our sister chromatids become their very own chromosome by the end of this process. So one more look at those DNA replication. Here's our strands. So you can see one there, and then the other is the blue one. And we split those apart, and an enzyme here, that's what this is going to be able to add new nucleotides following the base pairing rules to that complementary strand to make an exact copy of each. So that's really the beauty of DNA. So when you see one strand like this, it's kind of like an open book. If you're going to make a copy, you have to open a book up and look at it. That's what's happening here. And this continues until each entire chromosome is replicated. Remember, you have 46, so this has to happen very quickly inside of your body, because remember, it only takes a few hours for 24 hours about for a cell to completely divide, and DNA replication is only a portion of that time. So let's take a closer look. Steps of DNA replication. Here they are. So we're going to look at a couple enzymes and kind of look at this process step by step. So step one, an enzyme called helicase, so ace meaning enzyme and helicase. So helix, what does that make you think of? I'm thinking helix. And since the helix is really, or the helical shape refers to DNA, helicase unzips that. So really what's happening is this molecule is moving up this strand and it's splitting it apart by breaking these H bonds apart that are in between the bases. So this might be an A, this might be a T, and they're being broken apart. So we have to kind of open the book before we replicate. So those hydrogen bonds are getting broken down, and that's helicase's job. It's kind of like a zipper. It's going to unzip the DNA, and that's an enzyme. And then we get this fork shape. That's a replication fork that we're talking about there. This fork in the road there. So another enzyme, or really two of them, will come along and they're going to start rebuilding the complement, using those complementary strands as a template. So what's happening here is our helicase, we'll just put in, is still unzipping, but then these new polymerases are coming, and polymerase means it's an enzyme, but it's making that polymer, and using the old DNA strand there, the complementary side, it's making a new strand, this green you see here. So by it I mean the enzyme, and it's just adding nucleotides that are floating in the nucleus, and they get put in to make these new strands. So just to look simply, it's DNA polymerases add complementary nucleotides to each strand. So they're rebuilding two new molecules, one here, from the original. And these are exact copies, don't forget. That's the beauty of DNA and how it works. Finally, that process continues and we get two DNA molecules forming that are identical to the original molecule. So basically that original molecule, you can see the sugar phosphate backbone here, a new one was added by those enzymes and we end up with two identical strands. So the information here is the exact same information here. So these are exact copies of each other thanks to that complementary pair of backbones. And we call this semi-conservative replication, meaning one old strand is used to make a whole new molecule by adding a new strand. So it's called semi-conservative replication, using those base pairing rules. Remember, quick quiz, what pairs with A? Oh, too slow. T. What pairs with G? C. So adenine with thymine, guanine, and cytosine. And the other way around too. Oops. So looking at this all in one, we have this helicase unzipping the molecule. Process continues. We add new nucleotides to the complementary strand, and we end up with two new identical strands there. So here's a look at that, if I can get my mouse to work here. Let's see if this works.
DNA replication is defined as the process by which an organism's original DNA is used as a template for the production of a new complementary DNA strand. An enzyme called helicase unwinds the original DNA's double helix, creating a replication fork. Next, an enzyme named DNA polymerase III works down the leading strand and up the lagging strand of the replication fork, synthesizing two new strands of DNA by taking free nucleotides and pairing them with the complementary bases on the original DNA template. The process of DNA replication is described as semi-conservative because the two copies of DNA produced each contain one strand of the original DNA and one entirely new DNA strand.
So hopefully my friend there helped describe this to you. And here are our objectives again, just so you can use these to study. So our first objective was to describe the experiments of Watson Crick, Rosalind Franklin, which led to the structure of the DNA double helix, a very important discovery and probably one of the all-time most important discoveries of man. So don't forget that. DNA is probably one of the most unique and incredible molecules we've ever discovered. But don't hold me to that. As a biologist, actually, I'm 100% with that. So describe the structure of DNA and list those 4 nucleotides. What are they? Adenine, thymine, guanine, and cytosine. Remember how they pair, remember that these join together, they make those long polynucleotides, or the long DNA strands that we see. DNA is a very long molecule. If you took all the DNA in your body or really in one cell and strung it all out and lined it up, it'd probably be taller than you in just one cell. Remember, you have about 100 trillion of them. And they all have 46 sets of chromosomes, except for a few. Understand the base pairing rules, so the idea that adenine bonds with thymine and guanine bonds with cytosine. And we want to talk about the enzymes involved. So you had helicase and polymerase, remember what they do, and DNA replication. This is how organisms are able to make copies of themselves at the molecular level. So make sure you study these and are familiar. So long. Hopefully that was helpful.